All right, good morning everybody, or afternoon now I guess it is. Welcome. We're very fortunate today to take advantage of Ian Head, who is passing through on his way to a meeting down around Monterey. And when Ian mentioned this to me, I immediately invited him to come here to give a talk in Berkeley. Ian hails to us from Newcastle University, where he's a faculty member in the School of Civil Engineering and Geosciences. Ian's research is focused on the deep biosphere and on microbial hydrocarbon degradation in oil reservoirs. And together with Steve Larker from Calgary University, or University of Calgary, over the last 10 years, they've really created a paradigm shift in our understanding of microbiology in these oil reservoirs and the role that these microorganisms play in the formation of heavy oil. So it's a great pleasure for me today to introduce Ian and to hear his talk on When Worlds Collide, the Biosphere Meets the Geosphere in Deep Oil Reservoirs. So, Ian.
Thanks very much, John. Thanks for inviting me and taking advantage of me having to make a terrible trip to California. So, one of the most terrible places in the world to be. Sunshine, beautiful landscape, wine. It's been a real stressful few days, but nice anyway. Yes, I think John has very much sort of introduced my title slide, I guess. There are a number of people on here who are postdocs and PhD students through the years who worked on worked alongside us on anaerobic hydrocarbon degradation and also trying to integrate some of that information into a geological context. As we said, we've worked very closely with Steve Larker in the University of Calgary, and for me that's been a fantastic collaboration and really the multidisciplinary aspects for me as a microbiologist of actually getting some feel for the broader significance of the work in a geological context. Very valuable, I think, has allowed us to come up with some interesting ideas that I think otherwise we, as microbiologists working on our own, we might not have done. Always put in the acknowledgements of our sponsors at the start of the presentation, because I probably forgot that I have to do this by the end, so let's get this out of the way now. So over the last 10 years or so, we've been funded by a consortium of oil companies under the BACUS program. There have been 3 different phases of that, and the final phase involved these characters here, but we've also been fortunate enough to be funded by UK government agencies and taking advantage of things like the Federation of European Biological Society, have various short-term fellowship programs, and we've had various people over to work on some of the research on these themes, which has been very, very valuable. Okay, so what I'd like to do first of all, I guess the start's probably going to be a bit of telling you what you already know to some extent, but it's useful to put things in context. So I'll say a little bit about heavy oil and how that fits in with our understanding of the deep biosphere. Then start to think about petroleum reservoirs as a microbial habitat. Why might we have some interesting microbiology going on in these kind of systems? And then begin to look at some of the conceptual models that have been developed to explain the occurrence of heavy oil and how they've been revised over recent years, and also what microbiological processes may actually have been important in leading to the formation of heavy oil. And then go on to have a look at oil-water transition zone, which we think is probably one of the microbiologically most active parts of the petroleum reservoir microbiome, and maybe a hotspot in the deep biosphere, but we'll see that may not actually be the case. And finally, on the basis of all this, to look at, we've had a reappraisal about what does the petroleum reservoir microbiome look like, what does it mean for heavy oil formation, and some of the recent data we've been obtaining, which has made us think again about the system and question whether we really know as much about the system as we thought we did. And then if we've got a bit of time, so an interesting little anecdote, if you like, at the end about sort of how the research that you do can sometimes have impacts more broadly than you might at first expect. Yes, back in 1998, Barney Whitman at University of Georgia came up with some slightly more than backward in book calculations to estimate where a prokaryote biomass was on the planet. And a few startling things came out of that. First of all, they estimated that the total amount of prokaryote biomass was very close to the total amount of plant biomass on the planet. And then from the point of view of the deep biosphere, it turns out that most of the prokaryotic biomass is in deep subsurface sediments. The concentrations of cells are pretty low, but the extent of that particular habitat is huge. And when you multiply up to that, then you get some really sort of quite remarkable numbers. And the bulk of microbial life does seem to be in deep subsurface sediments. So the deep biosphere is probably the largest biome on the planet. This has been revised in recent years. Some really nice work from Jens Kallmeyer and Steve Daunt, where they've used a modelling approach to actually estimate how much biomass you should actually have in subsea floor sediments, depending on the productivity of the overlying waters. On the basis of that, these numbers have been revised down. So perhaps microbial prokaryotic biomass is only about 10% of the total biomass on the planet, but it's still the deep biosphere is still a huge biome. So although we're down by about an order of magnitude, it's still estimated at about 10 to the 20 times cells or so, prokaryotic cells in these deep subsurface sediments. So that's a pretty remarkable microbiological environment. Okay, so in terms of petroleum systems, how do the biospheres fit in here? Biosphere fit in here? And I would say that biodegradable petroleum reservoirs are actually probably one of the most obvious manifestations of the deep biosphere. And it turns out that most of the world's petroleum is not really in the Middle East. Most of it is in places like northern Alberta in Canada and also in Venezuela. But the difference with oil here from the oil in the Middle East is that it's heavily biodegrading. And whereas the biggest of conventional oil fields in the Middle East probably represent sort of hundreds of billions of barrels of oil. It's been estimated that there are several trillion barrels of oil in heavy oil fields. So the heavy oil inventory really outweighs the conventional oil inventory on the planet by some margin. And the reason we're here is because of biodegradation in the petroleum reservoir over geological timescale. So this is really a very strong indication, if you like, of the significance of the deep biosphere. We've worked mostly in the Canadian oil sands in northern Alberta, near Fort McMurray, for example. And this is just a little map of North America with Alberta highlighted. The white patches, sort of ironically, are the oil patches with Peace River here and the Athabasca oil sands. To give you an idea of the extent of these oil deposits, it's been estimated at about 108,000 square kilometres in extent, aerial extent, and that's slightly larger than the area of South Korea. So this is a really massive deposit of oil. Again, to see that in a very dramatic way, this is a photograph of an oil sands mine near Fort McMurray, operated by Syncruise. And just to sort of stand on the edge of this huge hole in the ground where they actually mine the oil sands because the reservoirs are very close to the surface. And to imagine that all of this mass of tar and heavy oil that's in these sands is there because of microbial activity. I guess the power of the small. Okay, so heavy oil is very widespread. It's I guess becoming a more important part of the energy mix as conventional oil reserves are beginning to dwindle. So how did they actually come about? And so there have been a few conceptual models developed to explain the occurrence of heavy biodegraded oil. And for many decades in the oil industry, the prevailing view was that the oil in heavy oil reservoirs was heavy because it was biodegraded aerobically by aerobic hydrocarbon degrading bacteria. And this is completely understandable because at the time, everyone was unaware of the ability of microorganisms to degrade hydrocarbons of crude oil under anoxic conditions. And so the emerging view of a petroleum system, if you like, is you've got a source rock where you've got organic rich rocks burning to temperatures where they start to thermally decompose. The oil migrates, it gets trapped by some impermeable rock layer and accumulates in a petroleum reservoir. So I guess if you like, that's the source term. The distinct term in terms of biodegraded oil is that you have a connection between an aquifer associated with the reservoir, and this is delivering meteoric water saturated with oxygen down to the reservoir, with aerobic hydrocarbon degrading bacteria. Eat up all the nice long chain alkanes that we like to turn into gasoline and other useful things, and basically the alkane more or less completely removed by the biodegradation process in this horrible, less valuable, sometimes sulfur-rich heavy oil, and the culprits were always thought to be aerobic hydrocarbon degrading microorganisms. And so at the time, you'd see quotes in that this is a sort of classic treatise on organic geochemistry, which has got some work on heavy oils. And they would say that anaerobic bacteria can live in the metabolites produced from aerobic hydrocarbon degradation, but do not grow on hydrocarbons. So this was very much the view. Shortly after this, Fitzgerald's group and also some work from John's group and other patients started to demonstrate that anaerobes could in fact degrade hydrocarbons in the absence of oxygen, and some very exciting and novel biochemistry that come out of this. The whole range of them. Okay. So sort of coinciding with the time we started to understand that you could get anaerobic hydrocarbon degradation, the conceptual model based on aerobic degradation of oil and petroleum
now being connected to the conference. Please stand by.
The amount of oxygen required from the surface down to the reservoir, which may be hundreds of thousands. In the context that in organic rich sediments, oxygen basically is consumed within millimeters of the surface of the sediment. You're also going to be transporting water with oxygen through rocks which have got reactive mineral species. So sulfide minerals, for example, oxygen will react very readily with those and be removed. So some problems with this conceptual model started to arise. So then once the question had been asked and then people began to give the answer, actually you probably have oxygen down to the petroleum reservoir to explain heavy oil by aerobic degradation. People started to look for evidence of anaerobic processes of hydrocarbon biodegradation in reservoirs. Some of the first evidence that suggested that in situ petroleum reservoirs you may be getting anaerobic hydrocarbon degradation came from gas isotope data. And so basically what this shows is the delta 13C of methane versus the delta 13C of ethane from two hydrocarbon gas. And typically as petroleum is generated from the source rock, you get cracking of the C12 bonds initially, and once they're all removed, if you like, then the carbon 13 bonds are broken. And so what this leads to is a situation where oils with low thermal maturity have relatively light methane, associated with relatively light methane, and as the source rock more and more oil and isotopically heavy methane. So what was observed in a number of biodegrading reservoirs was that the isotopically light methane associated with isotopically heavy methane, suggesting that this is a secondary biogenic gas that may be produced during the formation of the biodegraded heavy oil. And this is perhaps the first kind of in situ evidence that methanogenesis, for example, might be the formation of heavy oil. At this time, no one had ever demonstrated that hydrocarbons could cause degrading under methanogenic conditions. And then in 1999, Carlton Zengler demonstrated for the first time the methanogenic conversion of hexadeciane into methane. And so this, I guess, gives you the microbiological grounding that methanogenic degradation of the reservoir might actually be biological, as well as having circumstantial evidence from gas isotopes in the field. Not long after that, it says Steve Laughter and Martin Jones in Newcastle and the PhD student Carl Nathan started to look for the metabolites that identified as characteristic of anaerobic hydrocarbon degradation pathways, and they started to find these biodegraded heavy oil reservoirs, but not non-degraded reservoirs, and not in situations where oil was degraded aerobically in the laboratory. For in situ anaerobic degradation of hydrocarbons in situ controlled reservoirs. We then went on to I guess, follow up on the work of Carsten Zengler, and rather than looking at this pure hexadecane, we began to look at whether you could biodegrade crude oil under methanogenic conditions, and after a long wait and about 700 days, the experiment lasted, so we got rid of all our crude oil alkanes and converted them to methane. Fortunately, we were able to do this in the context of our joint industry project, where we didn't have to worry too much about the sort of 3-year life cycle of a PhD student, where this might have been quite one figure in your thesis. So in these systems, we saw essentially stoichiometric conversion of alkane to methane. We do the GC analysis. You can see the alkanes are all gone, and so during this work, we did a comparative analysis of methanogenic oil degradation, but also oil degradation under sulfate-reducing conditions, and tried to see whether there were any patterns in the removal of the hydrocarbons that might tell us something about whether this matched, or whether any of these processes matched up with what we see in real time. So what we've plotted here is just data from these experiments, NC17 to pristane ratio as a proxy for saturated hydrocarbon. Methyl biophenol to 4-methyl biophenol ratio is a true proxy for aromatic hydrocarbon degradation. And what we see with the blue dots here, sulfate-reducing systems, and you see that you get concomitant with alkane degradation, you get aromatic degradation as well. Under methanogenic conditions, alkane degradation, but we don't see degradation of the aromatic compounds here. Now, we also had a suite of oils from the North Sea, from the Gulf Fax field, and the Gulf Fax field shows a gradient of biodegradation, with some of the wells having very biodegraded oil and some having much less biodegraded oil. So we plotted the same parameters from these field-degraded oils, which shows that the field-degraded oils basically followed the same trajectory as the oils which were degraded in the lab under methanogenic conditions. So the methanogenic oil degradation in reservoirs a reasonable explanation for heavy oil. In terms of microbiology of these systems, what we observed was we got a very strong selection of organisms related to Syntrophus and Smithella. It's actually Smithella. Carsten Zengler identified the organisms in their system as they were actually Smithella. So they were very highly enriched in our systems. Another interesting observation was that the methanogens that were enriched were primarily CO2. Although we did get some enrichment for plastic methanogens, that was much less than the enrichment of hydrogen oxide and CO2. Led us to think that maybe syntropic acid oxygen in methanogenic oil degradation, so we'll come back to that. The experiments also let us estimate doubling times for these organisms, and it turns out that for the Smithella growing on the crude oil in the methanogenic consortia, the doubling time was something around 36 days. So these are pretty slow processes, but still much faster than the rate of biodegradation in situ. Okay, so that's some, I guess, some lab studies to inform observations that we can see in petroleum reservoirs. So what if we go back to the petroleum reservoir and use some of the information from the microbiology and see if that matches up with what we see there. So, for example, I mentioned that an indication that syntropic acetate oxidation might be important, that since we have a very high proportion of CO2-reducing methanogens in our methanogenic oil degrading system, and based on the stoichiometry, or the conventional stoichiometry of alkane degradation, we would expect a much higher proportion of plastic methanogens. So one way to explain this would be that, in instead of the acetate being converted directly to methane by plastic methanogen, it's converted first of all to hydrogen CO2, which is then used by. In this case, again, we're back to some gas isotope data from some field analyses, and looked at the relationship between the delta 13 of CO2 and methane in these systems. And Mike Whittaker has done a huge amount of work looking at the isotope schematics of methanogenesis and the number of domains which are characteristic of either CO2 reduction, classic methanogenesis, and also methane oxidation. And so we started to plot some of the gas isotope data from biodegraded petroleum systems and non-biodegraded petroleum systems. You can see that separately. This Steve Larcher's group in Calgary did some Rayleigh fractionation modelling to see what would happen if you took some alkanes from a crude oil, you oxidize them to hydrogen, CO2, and acetate, and then you either took your acetate and you channeled it down through acetate oxidation, or you use a superplastic methanogenesis. And this also involved mixing of the produced biogenic gas with any indigenous gas that was in the system. And so what we saw is if we had a system which had purely classic methanogenesis, the model was absolutely nothing like the isotopes we see in the biodegradable. Whereas we've got a lot of methanogenic CO2 reduction, it has to start to fit the observed gas isotope data much more clearly. And in fact, the best fit seems to come if we have the model run with 75 to 92% of the carbon is channeled through syntropic acetate oxidation, followed by methanogenic CO2 reduction. Very positive values for the delta 13 CO2, which seems to be something which is very characteristic of biodegrading petroleum reservoirs. It looks like, based on the fuel data, that this process whereby you have hydrocarbon degradation to hydrogen, CO2, and acetate, and then syntropic acetate oxidation, feeding a community of CO2 reducing methanogens, looks like this again might be what's occurring in the petroleum reservoirs. Another nice thing about working with the geologists, to take you out into the field, this is in fact the Athabasca River, and they like to show you outcrops, so geologists like outcrops, and I never really understood this until they took me to the Athabasca River and said this is an outcrop of what you're actually looking at. You're looking inside a petroleum reservoir. So the Athabasca River basically has cut through the oil sands deposits, and so what you see is shales and silts, which are essentially the cap rock of the oil reservoir, underlain by oil-saturated sands, which are the oil sands reservoirs. And underneath this, in this particular case, there's the Devonian carbonate and the river, so basically the aquifer. So this gives you a sort of a nice view of what it's like inside a petroleum reservoir. And based on this, plus some geochemical data, we began to think about how might a petroleum reservoir look to microorganisms. Where might they want to live in a petroleum reservoir? And this was informed to some extent by some work we'll be doing on Chinese petroleum reservoirs. And this is just sort of the percentage of saturated hydrocarbons in these reservoirs as you go from the top of the reservoir to the bottom of the reservoir. You start off with about 40% saturated hydrocarbons down to about 20 or so percent near the base of the oil column, where the oil column basically sits on top of the aquifer associated with the reservoir. And this led us to suggest that this oil-water transition zone was probably the site of the focus of microbiological activity in biodegrading petroleum reservoirs, where you've got the microbial community being fed by diffusion of hydrocarbons from the oil column down to the oil-water transition zone, and things like inorganic nutrients, for example, or if it's a sulfate-reducing system, sulfate being supplied by the aquifer associated with the reservoir. And some other observations that tend to support the fact that this oil-water transition zone is a hotspot for biodegradation is in situations where you have a reservoir which basically has an underscale as well as the top, and so you don't actually have an aquifer in direct contact with the oil tend not to be biodegraded. So I guess we're beginning to develop an idea of how a petroleum reservoir might look as a habitat for microorganisms. That particular reservoir was in China. It was biodegraded, but it was relatively moderately biodegraded. Most of our work has been done in the oil sands in Alberta, where the oils are much more heavily biodegraded. And so we asked the question: in these very heavily biodegraded oils, do you still see the signal of microbial activity, the oil-water transition zone? These data show what's called a resistivity log. Basically, resistivity is a proxy for hydrocarbons in the sediment. And so up here, you have high resistivity, you get high hydrocarbon saturations, and down here towards the water transition zone. You've got lower hydrocarbon saturations. So again, in this case, it looks like we see these gradients in the oil, from the oil column to the oil-water transition zone. And from this system, we were able to get core samples and actually characterize the hydrocarbons in terms of the saturated hydrocarbons, aromatic hydrocarbons, and also the polar fractions, the resins and the aspartame. These oils, as I said, are much more biodegraded. You can see the saturated hydrocarbons with the red symbols here are at a much lower level than they were in the Chinese reservoirs. So there's a very slight gradient here. Where you do see the gradient are in the aromatic hydrocarbon fraction and also in the aspartame, so in the opposite direction. It's a typical biodegradation signal where you get decrease in the hydrocarbons, decrease in the fractions of biodegradation. We're also very fortunate with this set of samples. I guess the guys here that work with BP on the petroleum reservoirs know how difficult it is to get good samples for microbiological analysis from petroleum reservoirs. And although we worked for the oil companies for 10 years, we had a very small number of really good field samples from them. But in this case, we struck lucky. This comes from the oil sands. They're actually drilled quite frequently compared to certainly more so than offshore locations. But also you have the advantage that in northern Alberta, because the oil's heavy, they use something called steam-assisted gravity drainage to actually melt the oil to make it easier to produce. And they need accurate water contents of the oil sands to know how much heat they have to put into the system to actually recover the oil. And to do that, they take a core and freeze it immediately so that they can then take it back to the lab and get accurate water content. So we were fortunate to get core samples which went through this oil-water transition zone, which again is something which oil companies don't want to do. They don't want to be drilling into an aquifer. They want to produce oil and not water. So we're very fortunate to get these samples. We got the hydrocarbon analysis done. We were also able to extract DNA and introduce some microbial community analysis. And in the first instance, we did some qPCR to quantify the bacterial populations. And interestingly, what we see is that the oil-water transition zone, we do see an increase in the bacterial populations of 2 to 3 orders of magnitude compared to the zones outside. So this was the first sort of evidence, if you like, that there was a hot spot for the biology at this oil-water transition zone. We began to look at these data in the context of, if you like, the broader marine deep biosphere data that are out there from the ocean drilling program, for example. There's thousands of data points being collected to count prokaryotic cells in deep subsurface sediments. Although the oil sands are currently in a terrestrial environment, they're actually marine sediments that when they were deposited, and so it's probably reasonable to plot these data on this marine sediment data set from the Interestingly, our oil-water transition zone samples plot sort of pretty much where you predict on the basis of this trend line, whereas those in the oil column have got much lower numbers, and these kind of numbers of cells you see in like normal deep subsurface sediments usually are associated with low productivity environments and or stressed environments. But the interesting thing is that in the oil reservoir, where you get lots and lots of electron donor, you might expect it to be a real hot spot in the deep biosphere. Actually, probably not. It's pretty much like the rest of the biosphere in terms of cell numbers, so there's probably not more energy available to these organisms in these systems that might reflect the fact that they're living in an environment where you've got lots of toxic chemicals and like the solvent properties of oil. So the oil water transition zone is definitely a hotspot in a petroleum reservoir, but not in the biosphere. Right, I've got this here because I'm afraid there's about to be a slide which is a bit old school and might not be a bit sensitive about what I'm going to show. It's actually it's a DGG gel. Okay. But I'm not going to apologise for that because I do think DGG is actually still a very useful approach, and just because you can do super deep sequencing doesn't alter the fact that you can still get useful information, albeit lower resolution from this kind of analysis. So for these samples, we did some DGE analysis, and it showed things were not very exciting. There's not many different organisms. There were just a few things dominating the community. But what we did see was quite interesting, and that the most dominant organisms were related to Acinetobacter and Pseudomonas, and particularly the Pseudomonas seemed to be selected at the oil-water transition zone. So this was a really interesting data set, and we sort of hid this away and sort of worried about it for quite some time because it just didn't look like anything we expected, especially in the context of the emerging model of a methanogenic oil degrading system with Smithella and methanogens and not Acinetobacter. So these organisms they're obviously well known for their metabolic versatility. They're known to degrade a huge range of different compounds, including aliphatic and aromatic hydrocarbons. But anyone who reads the microbiology textbook will tell you Acinetobacter and Pseudomonas are basically aerobic heterotrophs, and maybe they can use nitrate and electron acceptor as well. So, we have a bit of a dilemma. What are all these aerobes doing in a petroleum reservoir? The reservoir is about 600 meters deep in the oil sands, just to give you a bit of context for that. So I just thought I'd show you that again. It is a DGG gel. Okay. Yeah. So clearly we asked ourselves, these look like what we expect to be aerobic organisms, but what actually is the metabolic potential in the reservoir. So we did some metagenomics. We had to use some whole genome amplification. Relatively small data sets, but it was a couple of years ago that we did this. But still enough to give us some useful information. First of all, if we just did a sort of phylogenetic categorization of the reefs, the bulk of the reefs were bacterial. There was very few archaeal sequences in there, which is interesting. But what was also interesting is that we could really very clearly see a distinct difference in the communities at the oil-water transition zone and the oil column, simply just by looking at the GC content of the DNA. That was quite interesting. That there's information even in the sort of the humblest parts of your metagenome as it were. Okay. So again, we started to drill a bit deeper into the potential source organisms for the metagenome reefs, both in the oil column and the oil-water transition zone. Excuse me. The system was dominated by proteobacteria, which makes sense with Acinetobacter and Pseudomonas. Drill down a little more deeply. Again, both dominated by gamma proteobacteria. The oil-water transition zone, they're not quite so dominant, and we see more alpha proteobacteria and beta proteobacteria. And then if we go slightly higher resolution again and look at the composition of the reefs from the gamma proteobacteria, they mostly turn out to be from the Pseudomonas aeres, and mostly from the Moraxellaceae and the Pseudomonas aereae. And you drill down to genus level, Acinetobacter and Pseudomonas are what drop out as you might expect from the DGG analysis. Okay. But in addition, as we saw in the DGG analysis, the oil-water transition zone, we seem to have a higher prevalence of Pseudomonas than there was in the oil column. Again, that's reflected in the reads from the microgenome. Again, I guess reassuring this to some extent if you actually go and look at the GC content of the genomes of Pseudomonas species. They correspond to about 62%. GC, which corresponds to this peak where we have more Pseudomonas in the system in the oil-water transition zone, and Acinetobacter genomes are about 40%, G+C, which matches up with this second peak in the oil column sample. Okay. We then look at this, if like the metabolic characteristics of the reeds at a very broad level. We categorise the reeds into those that look like they came from aerobic pathways, anaerobic pathways, etc. And we compared those with metagenome data from methanogenic oil-degrading microcosms, which we knew genuinely were methanogenic oil-degrading systems. And you can see in the methanogenic oil-degrading system, the bulk of the reeds in this analysis come out with anaerobic pathways, whereas in the two oil sands core samples, the majority of the reeds come out as matching up with aerobic pathways. Well, there's still a reasonable proportion of anaerobic pathways detected. Again, if we look in a little more detail, this is looking at the KEGG xenobiotic metabolism pathways and the reeds that are assigned to those. I'm going to focus on two pathways here specifically. Benzoate degradation, because there are two sort of well-characterized pathways, one which is aerobic and one which is anaerobic. So the benzoate ligation pathway is the anaerobic pathway for benzoate degradation. You see that's very prevalent in the methanogenic oil-degrading system, and less so in the oil sands core samples, but still a considerable proportion of the reeds that map to categories. And again, for the aerobic pathway, benzoate hydroxylation pathway, you see them more represented in the oil column and the oil-water transition zone samples than they were in the true methanogenic oil-degrading. Again, also, as you might expect, we find a full suite of fatty acid metabolism genes, which we'd expect to find for something which is driven by alkane degradation, for example. But in addition, we also found a number of reeds which mapped onto alkane monooxygenase like genes as well. So it looks like there are genuinely aerobic pathways in these. To say this was an unexpected observation that not only the phylogenetic analysis but the metagenome analysis suggests that we've got aerobes and metabolic pathways that require oxygen in these heavy oil reservoir core samples, and are dominated by Acinetobacter. Okay, so why might that be the case? I guess our first thought was, oh my God, we've gone terribly wrong, and we've got the oxygen into the samples. These guys have grown up, and it's nothing to do with what's actually in the reservoir. So that's the first possibility. During sampling and transport, we got oxygen into our samples. So definitely that's a possibility. And well, we wanted to be sure that what we saw was real. We started to think about what signals might we see if this was truly oxygen ingress and then growth of aerobes in our samples, mapping through in situ population. First of all, we might expect growth yields on oxygen to be much higher than they would be in a methanogenic system. So even a small amount of oxygen getting into the system could lead to overgrowth of your indigenous community by aerobes. Okay, but if that was the case, you'd perhaps expect to see the same community selected all the way across. But we do see differences as we go through the oil column, through the oil-water transition zone. We do see what appears like selection of Pseudomonas at the oil-water transition zone. So why would it be so systematic? Also, why are the populations so low? We see that tends to treat cells for lots of organic carbon in these systems. If you give them some oxygen, there's no way you're going to maintain populations that low. So that's a few things that look a little bit inconsistent. But you might expect to see small numbers if you've got limiting oxygen concentrations and because of the high maintenance requirements for maintenance energy requirements of aerobes, then under limiting conditions, you might not expect to see massive outgrowth, if you like, of any of those techniques. So it's a little bit up in the air, shall we say. The other problem with this was that it's not possible to go back to our samples and say, okay, this is what we have. We've got a system dominated by Acinetobacter pseudomonas. What was it like before they appeared? Because we can't get those samples because we've got what we've got. So instead of that, what we thought was we'll make the assumption that these are really the organisms that were there. So let's see what happens if we do actually thaw out our cores and expose them to oxygen. What happens to species? Do they look like they're still dominated by pseudomonas and Acinetobacter? So we did that, and so in our original samples, this is just basically taking the leaves which were more than 1% of those detected. Again, so we obviously see Pseudomonas aeries are the dominant organisms in the oil column samples, or water transition samples. But if we take some of the core sample, thaw it and expose it to air, we see the community changes really quite dramatically. So we do have Pseudomonas aeries, but their representation is way down. And a massive increase in these actinomyces aeries, which actually turn out to be something called biosphere, which is an aerobic hydrocarbon degrader. so when we do deliberately expose the samples to oxygen, we do see rather large changes in the community, suggesting that oxygen exposure, while we've been handling our samples, has not been the issue that the organisms being dominant. So maybe they are actually the main source in there. Other possibilities, maybe these guys are aerobes in there and they're getting oxygen which is being generated in situ. Again, that's definitely possible. I guess the same arguments about sort of what might you see if you got some oxygen into the system. But the question arises, where would oxygen come from in these systems? In the deep biosphere, radiolysis of water has been looked at as a source of hydrogen and energy to drive the deep biosphere, and it looks like that is certainly feasible in certain circumstances. That's obviously going to be dictated by in situ radioactive decay, and if you have reasonably high levels of radiation, and maybe you could also produce enough oxygen that might feed into it. We did some calculations looking at the rates of oil degradation relative to the rates of oxygen generation you might expect to see. We're quite conservative in that, and even if you let the oxygen be produced at the maximum rate possible from radiolysis, it would only be enough to account for about 1% of the degradation of the oil that we see in a heavy oil reservoir. So in terms of the quantitative process leading to the degradation of the oil over geological timescale, it's probably not feasible, but it doesn't necessarily mean that it's not something which, if you like, locally on more recent timescales might have a role. Other sources of oxygen might be dismutation of nitrogen oxides or chlorate. I only used to discount this one until yesterday when John told me they started to find chlorate in chlorate reservoirs. So maybe there's some chlorate down there that could, in principle, lead to the formation of small amounts of oxygen that could feed aerobic system. Another possibility, at least in the oil sands, is that maybe it is oxygen meteoric water as the original conceptual model suggested, and that could be facilitated by what I call isostatic pumping of oxygenated water during glaciation cycles. So in northern Canada, over the last sort of tens of thousands of years, there have been various cycles of glaciation and deglaciation. So essentially, when you're in a glacial period, you'll have lots and lots of ice sitting on top of Alberta, squashing down the oil sands reservoirs. Once that starts to recede, you get the pressure is removed, you get isostatic rebound. The reservoir sediments are essentially like a sponge, and they expand as the loading comes off them. You get lots of meltwater, which is rich in oxygen. These are relatively shallow reservoirs, a few hundred meters, so maybe you've got circumstances that might deliver some oxygen down into these reservoirs. Though again, this is under the lifetime of these reservoirs, and it's probably not a significant source of the formation of the heavy oil itself, but it might explain why now we might see an aerobic population. The final possibility is that these organisms, which we are assuming are aerobes, actually might be anaerobes, which again, sort of, it goes against everything we're taught in the microbiology textbook, but actually it's something we should consider. And interestingly, there have been more and more publications coming out recently also demonstrating that there seem to be lots of aerobic organisms present in petroleum reservoirs. I guess this is the highest profile one is this one here from Gerrit Rodel's group in Calgary, as part of the hydrocarbon metagenomics program, where they analysed a large number of different samples from hydrocarbon resource environments and shown a large proportion dominated by aerobic and aerobic classes. Okay, so what does this mean? We have to revise everything we've said and go back and say, okay guys, you said it was oxygen, we said, well, it's not going to be oxygen, let's find a way to degrade oil in the reservoir anaerobically. We've got some mechanisms that will let you do this, but actually, is that now all wrong as well, and do we have to revisit the original aerobic degradation paradigm? Well, again, if we actually think in terms of the large scale, is aerobic degradation likely to have led to the formation of the huge deposits of heavy oil? I think the answer is probably no. So again, if we go back and look at the gases in these biodegrading reservoirs, they're basically dominated by methane with low levels of CO2. Again, the gas isotopes tell us we're isotopically heavy CO2, isotopically relatively light methane, which looks like a signature of methanogenic degradation of the oil. So in quantitative terms, it looks like the best explanation is probably still methanogenic degradation of the oil over. Also, if you start to sort of look around and see what can Pseudomonas really do that's not in the textbooks, so you sometimes go to some strange places, and this is a paper looking at the potential to produce hydrogen from wastewater sludge. And in this study, they isolated an organism, Pseudomonas species GZ1, using Hungate method, so basically a classic method for isolating anaerobes. And this Pseudomonas grows an organic carbon from either the sludge or sugars in culture, this pure culture, ferments sugars and generates hydrogen. So basically, in principle, it could act as a fermentative centropic partner with hydrogen. Perhaps the most striking case that I look at comes from Lisa Geek's group in Calgary, where again, they've been studying methanogenic oil degradation. They've got a culture which is enriched from a heavy oil field in southern Alberta, which basically converts both light oil and heavy oil to methane relative to oil-free controls. And it basically actively degrades crude oil alkanes quite effectively, converting into methane. Now, when Lisa analysed these cultures, as we've seen before and others have seen before, many methanogenic oil and hydrocarbon degrading systems are dominated by these Smithella. Again, that's what they found. Interestingly, though, Lisa also did some other experiments to try and begin to emulate what might happen in a real petroleum reservoir if you were going to develop an enhanced energy recovery method, for example. So if you use these cultures to inoculate sand columns saturated with residual oil, and shut the columns in for a year, measured methane production over time, showed that the oil was being converted to methane, and then after the end of this incubation, they analysed the microbial. This is what they inoculated with, dominated with Smithella. After a year of incubation in the sand columns, this is what the community looks like. The major bacteria were Pseudomonas in an actively oil-degrading, methane-producing system. So that's sort of, so I'm beginning to now think that maybe Pseudomonas, there's more to it than we originally envisaged based on our previous knowledge of it. So just to summarise, how does potential aerobic oil degradation in the Western Canada Sedimentary Basin, WCSB, fit with the broader landscape of oil sands, so the heavy oil in northern Alberta? The estimated degradation rates we estimate from compositional gradients in the oil, for example, and how that relates to geological measures of charging the reservoir, don't match up with an aerobic degradation pathway. The timescale would be likely to be much shorter. The CO2 and methane isotopes also suggest that we actually have long-term anaerobic, probably methanogenic degradation of crude oil in these systems rather than aerobic systems. Hydrostatic pumping of oxygen oxygen-rich glacial waters into relatively shallow reservoirs is a possibility, but that's only likely to have happened over the last tens of thousands of years. So not something which would have been, the oil sands have already been heavily degraded by this time. Heavy oil at this time. Mechanisms for the generation of oxygen indigenously are probably not enough to explain the degradation of the extent of our oil, the degradation that we see in heavy oil reservoirs. But it is possible that these kinds of organisms are aerobic latecomers who are basically living off the leftovers from the initial anaerobic hydrocarbon degradation over millions of years. Finally, maybe Pseudomonas and have latent anaerobic metabolic capacities which we're not fully aware of. Finally, I'll show this the same in Berkeley as it is in Newcastle. We're always being asked to demonstrate how our research is having impact above and beyond sort of academic impact. And so ourselves and a number of others have used sort of data like this, which shows you can convert crude oil alkanes into methane to suggest that this might be a means to enhance energy recovery from the stranded residual oil in reservoirs at any lifetime. So the idea is you pump in some nutrients and some magic dust, and you stimulate methanogens. The residual oil is converted to methane and CO2. The methane dissolves in oil, it gets more viscous or less viscous, makes it easier to produce, so you can produce more oil. Eventually produce enough methane that you repressurize the reservoir, produce more oil. Ultimately, volumetrics of production are much more favourable, and you produce more methane. So all very nice. And so you get shale gas and the bottom of the methane market. But that's one kind of impact. But I think the biggest impact that our work has had is actually cultural. So this book here, Petroplate, actually written by Amy Rogers, who's an associate professor in UCLA, and so this story is basically about a group in UCLA who are sponsored by a large oil company apparently, and they've genetically engineered some trophies to be really fantastically good at excluding hydrocarbons, just as we said, as an energy recovery vector. But all goes terribly wrong because engineered some trophies escape from the lab. First of all, there were explosions in La Brea, then storage tanks of gas stations get contaminated, they explode. The fuel line that's feeding kerosene to LEX gets contaminated. It goes into airplanes. Airplanes are falling out of the sky. Absolutely disastrous. So absolutely fantastic story. A real page turner. But of course, the real impact is not that. The real impact is that in the technical appendix to the book, our 2008 paper is actually cited as a source. So I don't expect a good thing or a bad thing. But the reviews of the book, I think, just say everything. And one of the reviews says that the science is utterly believable. So I'll leave it at that.
It's pretty impressive to impact pop culture. Ultimately, that's our goal in life.
$2 from Amazon.
So, are there any questions for Ian? Great talk. Ryan?
So Pseudomonas is a notoriously broad vector of genus. Do you get a feel whether you're isolating or whether you're mapping reads from across the genus, or are you getting a few strains? And if so, are there isolation efforts underway?
Yeah. Most of them read look like they map onto Pseudomonas stutzeri, so it's relatively narrow. But yes, we should try to isolate them. There's a slight difficulty with the nature of the samples that we have because they come to us frozen, which is great for doing the in situ community analysis, but not great for isolating things from. So we haven't got around to doing that yet. But I think actually we should try to do some fairly simple things like Pseudomonas selective agar, see what we can isolate, and then start to try and grow them anaerobically, as well as get some into the fungate tube.
How many meters do we have?
Trophy logic. I haven't yet, but it will be.
1,000 meters?
In one of the slides, I don't know.
Yeah, we see Pseudomonas.
And it's aerobe. Beneath oxygen.
We don't know. We find the genome, but it doesn't mean.
That's thermodynamically directing it all forward. But Pseudomonas that have never been shown to grow syntrophically, and they'll happily ferment, and they will happily produce hydrogen regardless as a result of fermentation. So do you, in this situation, do you think that it will be an obligate or a non-obligate syntrophy of the hydrocarbons?
I think I tend to think of these things mostly as non-obligate. I guess that the methanogens should, in principle, be able to use hydrogen and acetate from wherever it comes. But if there are other factors involved, so I guess that there's plenty of evidence that you need direct cell-to-cell contact. If there's something specific about that.
The top layer, they're less toxic, but I don't report it.
I guess where I was going with this is that if there was no methanogenesis in there, you should still see hydrocarbon degradation, and you simply see the hydrocarbon, the heavy oil should form regardless of methanogenesis.
Right.
In this system, you just build up more acetate, you build up more hydrogen.
I guess that when you do the thermodynamics for the hydrocarbon degradation, the hydrogen is sort of less than.
That makes it an obligate syntrophy, not
in that case, yeah.
Probably, yeah. Okay. How do you think pressure or temperature would impact this?
Yeah, almost certainly will. Pressure is a really interesting one, though. So I guess what you might think would be pressure, energy, as it were. So anything which reduces the volume of gas.
Amplification, the same thing as amplifying examples.
Should be more capable under high pressure than acetate being converted to methane and CO2. But the other thing is, I guess, overall, no matter what route you take, the stoichiometry is the same.
So whole genome amplification have several techniques. You can have rolling amplification. That means one enzyme just keeps going for life.
High pressure would be good for
and primers like PCR, they do sensitive regions. So it depends on the primer design.
Absolutely not. No, this is basically take your sample, smash it, get the out, and again, frozen samples not ideal for doing some microscopy, that kind of thing. So we're in some ways stuck by this. We have to
just do it naturally, like a rolling application.
Any other questions?
I don't think they find it enzyme that.
So lunch is served outside, and I believe because I'm the host, I get to go first with these. I'll have to wait. All right, thanks, guys.